and ejaculation. And you are going to pee in your pants? Not quite. It'll be a little stressful, all right, but it's actually not too complicated when you break it down to the basics. So nobody pass out as we move through this. Okay, you actually know more about this than you think you do. Having that warning and you're all scared, let's look at it. So we have essentially, all right, before we look at the details, this is a reflex. And in a reflex, you have sensory components and motor components, all right? So in the autonomic reflex of the pupillary constriction, light goes into your eye, activates the optic nerve, which is carrying sensory to the brain and that brain stem, and that reflexes with the outgoing motor via the ocular motor nerve to the smooth muscles of your iris, and your iris constricts. We got a sensory arm and a motor arm. Okay, we have that with any reflex. So erection and emission are primarily autonomic. Ejaculation is primarily voluntary, although the autonomic is going to be a component of it. So essentially what we're going to have when we have an autonomic pathway is we're going to see a sensory coming from the glans penis. It could be motor, it could be thought pathways, all right? So you're getting ready if you're a guy to ejaculate and provide the sperm, and so you watch that XXX movie or Playboy or whatever. So yes, that can be the sensory component. We're going to stick with the glans penis, okay? So sensory from the glans penis, as it leaves the glans penis, is traveling along the dorsal surface of the penis, and there it's forming the dorsal nerve of the penis. Okay? As it passes into the pelvic cavity, it's going to join other sensory nerves and motor neurons. The ones that we just talked about, going to the bulbal spongiosis, into the ischiocavernosus, into the external urethral sphincter. As it joins those, it then becomes part of the pudendal nerve. Other neurons joining it. It's like having a street that's called one Martin Luther King Boulevard and somewhere else it's called a different name. You're still on the same road, okay? And then it's going to carry this sensory information. Cell bodies are found in the dorsal root ganglion, and it's going to carry this information into the spinal cord. It's going into the sacral level of the spinal cord, okay, down near the conus medullaris. So this is sacral spinal cord. Erection, the dilation of the arterioles in our rectal tissue, is a parasympathetic function. Do we find parasympathetic neurons in the lumbar or the sacral spinal regions? Sacral, cranial, sacral. All right, so it doesn't have to go very far. It's going to form a synapse, an arc, with our motor parasympathetic neurons. How many neurons are in this peripheral nerve for the autonomic nervous system? 1 or 2?
2.
2. So the preganglionic neuron is going to come out into the pelvis. And then the postganglionic neuron is going to go to the blood vessels in the corpora cavernosa and the corpus spongiosum. Okay? So the preganglionic neurons coming out of the sacral spinal cord are called the pelvic splanchnic nerves. If you had me last semester, this should sound familiar. Okay? The postganglionic neurons for parasympathetic typically are found in the wall of the organ, like the bladder wall or the intestinal wall. In this case, they're not. They're found in the pelvic cavity. And as they travel into the penis, to the blood vessels, they are called the cavernous nerves. Okay? So the preganglionic are known as pelvic splanchnic, the postganglionic are known as cavernous nerves, but they're still parasympathetic. Okay? So that's a simplified version of the next slide that I'm going to show you. On this diagram, here's the dorsal nerve of the penis, which is carrying sensory. Joining with other nerves, it forms the pudendal nerve and goes into the sacral spinal cord. Okay? Coming out, we have the, where's the cavernous nerves on here? I thought they were on here. Over here. We have the preganglionic neuron, pelvic splenic nerves, and the postganglionic neuron, the cavernous nerve. Okay? I'm not going to diagram any other nerves different than that. It just looks more complicated. Okay? So, let me draw this on the board. And you have these on the canvas slides. Just trying to fit it on the board. Okay. So here we have the spinal cord. I'm going to draw it and then turn the lights off so we can see it. And I'll review the spinal cord with you. So this is the lumbar region of the spinal cord. Because we need sympathetic for emission, okay? This is the sacral region of the spinal cord because we need parasympathetic. So here is the gray matter of the spinal cord. Anterior gray horn, posterior gray horn, okay? Now up in the lumbar region, you'll notice we have a lateral gray horn, because that's where the cell bodies are for sympathetic. We don't have a specific structure for parasympathetic cell bodies. All right, then we have the spinal nerve structures forming the dorsal root. Here's our dorsal root ganglion. Here's our ventral root. And there's the spinal nerve. The spinal nerve has a dorsal ramus going to the middle of your back and a ventral ramus. Okay, bless you. So all of this is the ventral ramus of the spinal nerve, which carries both sensory and motor. Okay, so same thing up here. All of this is ventral ramus going out to typically the skin and muscles. And now we have the sympathetic chain, which although our sympathetic fibers are only in the thoracic and lumbar region, every ventral ramus has postganglionic neurons, because that's what goes to your skin. We talked about that with the dartos muscle was sympathetic innervated because it was out in the skin. Okay? So traveling along either side of the spinal cord, we have our spinal chain ganglia, which are known as the paravertebral ganglia. And they go all the way down into the pelvis. So as a review, for the parasympathetic, it has its cell body in the lumbar region. For the sympathetic. So its cell body is going to be in the lateral great horn. It's motor, right? So it's going to leave through the ventral root. So this is sympathetic preganglionic neuron. It's going to come into the sympathetic chain, and it can go up or it can go down. For our example here, we're going to just leave it where it is. The postganglionic neuron then leads through this. These are the rami communicantes. And this is going to go out the ventral ramus to the skin for sweat glands, or to blood vessels in the skin or skeletal muscle. So only sympathetic gets out to the skin. This is how we would get sympathetic to our dartos muscle, okay? To skin. All right? So, parasympathetic, notice how this one only has one ramus communicantes? That's because this has no sympathetic neurons. And so, if it comes down, instead of stopping here, we'll bring another one down. So there's no white ramus communicantes because there's no preganglionic neuron coming out of the sacral spinal cord. So we only see one of these at the sacral level. Okay? Now, parasympathetic are going to have their cell bodies here. In the sacral level, we're ignoring cranial like the vagus nerve and so on, because they're not getting to the reproductive organs. They are also motor. So this is going to be the green is going to be parasympathetic. Okay? It does not go to the skin. So it's only going to be in the ventral ramus for a short time. And then it's going to leave the ventral ramus to go into the pelvic cavity. So this is known as the pelvic splanchnic nerve. PS helps you remember this is parasympathetic. Splanchnic refers to viscera. It's going to an organ. And it can have multiple neurons. Okay? Now, it normally goes into the wall of the organ like the bladder, but in this case, it stops in the pelvis. And the postganglionic neuron goes into our blood vessels. And so these would be the cavernous nerves. All right, so I'm going to erase the sympathetic because they don't have a component to to erection. They do for emission. So we'll come back and look at it with emission, but we don't want it for erection. And then the organs that we're looking at, we have the penis. There's the bulb of the penis. And we have the urethra. The prostatic urethra, the membranous urethra, the spongy urethra. All right. We have the testis and the epididymis. And then we have the vas deferens. Wrapping, once it's in the pelvic cavity, laterally around the bladder, forming the ampulla, and then joining with the seminal vesicle to form the ejaculatory duct. Okay. I don't think I have a lighter green for that, so we'll go ahead and make that green. So here's the seminal vesicle, ejaculatory duct, prostate gland, vulvourethral. And then we have the urinary bladder. And we have our skeletal muscle. We're not seeing the ischiocavernosus because that's lateral, covering the crus. But here would be the external urethral sphincter. And here is the bulb bulbospongiosis. Okay? Almost ready to turn the lights off. Ejaculate for erection, okay? We have the dorsal nerve of the penis. Sorry, that's not on there. Let me draw that in blue. We have the dorsal nerve of the penis. In this area around here, it's going to change with, as we added other neurons, it's going to change and be called the pudendal nerve. Okay, so dorsal nerve of the penis. Here we'll call it the pudendal nerve. It has other neurons in it. It's going to carry that sensory information from the glans penis into the sacral spinal nerves. Cell body is going to form the dorsal root ganglion, similar to what I drew over there. It's going to go into the posterior gray horn, and then it's going to connect to the parasympathetic system for our motor pathway. And that would be the preganglionic neuron of the parasympathetic system, forming the pelvic splanchnic nerve. Then in the pelvic cavity, it's going to form it's going to synapse with the postganglionic neuron, forming the cavernous nerve. And that's going to go to the blood vessels of the corpora cavernosa, mainly, some to the corpora spongiosum. And so that's going to release neurotransmitters and cause the blood vessels to relax, and we have erection. Okay? So this is the pathway for erection.
I'm sorry to interrupt. What did you say about the relax?
So the release of the neurotransmitter causes the smooth muscles of the blood vessels to relax. They dilate, fill with blood, and that compresses the veins. And the penis becomes erect because of the enlargement due to the extra blood vessels, because it can't leave from the veins because they're collapsed due to the dilated arterioles. Okay? All right, so that sequence, the sensory part of our pathway is going to remain the same for emission and for ejaculation. Okay? Starting with the dorsal nerve, going to the sacrospinal cord, and then forming a synapse either with our parasympathetic for erection, our sympathetic for emission, or both, as well as lower motor neuron for ejaculation. Okay? So we're going to come back and look at this, but I want to talk about the biochemistry of the erection, the neurotransmitters. We're not going to worry about that for emission or for ejaculation, but I want to talk about how the arterioles dilate. Okay? So you have this image on canvas. I want to erase those neurons in preparation for emission, but I want to now, and then I want to talk about the physiology of erection, and then come back to emission and ejaculation. Does everybody have this down? Any questions about this sequence? Just think of it as a reflex. So there's a sensory pathway and a motor pathway. Okay? One neuron for sensory. Because it's autonomic, we have two neurons for the motor. Alrighty? So I'm going to leave the sensory nerve in place because we're going to talk about that for all three: emission, ejaculation, erection. Well, not in that order. I'll redraw these when we talk about ejaculation because we'll use them again, or the sap. Okay? All right. So this next step looks complicated, just like this one looked, but I broke it down and I think it wasn't as complicated as that diagram might have first appeared. Okay? You may disagree with me, but not as bad as it could have been. And the same with this. We're going to use some terms that you're already familiar with. So listen to me carefully as I break it down for you. All right? Already looks complicated. So, what was the calcium used for in skeletal muscle contraction? Why was calcium important for skeletal muscle contraction?
Contraction.
So when calcium came out of the sarcoplasmic reticulum, what molecule did it bind to? Troponin, similar to calmodulin. And when it bound to the troponin then moved aside. Tropomyosin moved into the actin groove deeper, and that exposed on the actin the binding site for myosin. Okay? The myosin then swung forward and pulled the actin over the myosin. That's what we are blocking from happening when we do erection here. All right? So parasympathetic neurons can do this in two ways. They can actually themselves, here's the cavernous nerve. Remember we just talked about the cavernous nerve? All right? So there's the cavernous nerve releasing a gas. You may not be aware, but gases can be neurotransmitters. They can move across the plasma membrane. So the receptors are actually inside the cell. So the cavernous nerve releases nitric oxide. We can also get nitric oxide by just regular postganglionic nerves from the parasympathetic system binding to cells that line the blood vessels, line the arterioles, and releasing acetylcholine, and that causes those cells to make nitric oxide. So the nitric oxide coming into the smooth muscle of the blood vessel can come directly from the cavernous nerve, or it can come from the epithelial cells that line those arterioles. Okay, so two different ways, but both of them end up forming nitric oxide. Okay, the nitric oxide moves into the cell and activates guanylyl cyclase. Remember hearing about guanylyl cyclase the first week of school? It wasn't a G-coupled protein. Okay, it was actually attached to the receptor. Guanylyl cyclase converts GTP to cyclic GMP. Sounding familiar? All right. Now, cyclic GMP is going to do two things. It's going to activate a protein kinase, all right, which is going to cause calcium to be pumped back into the sarcoplasmic reticulum. If we're pumping calcium back into the sarcoplasmic reticulum, what's happening to the level of calcium in the cell? It's going down, which means it can't bind to troponin, which means it's gonna, exposed sites of actin for myosin are gonna be covered up. So we're gonna have fewer myosin heads binding to the actin, which is what happens when a muscle cell relaxes. Okay? So this is one reason why this is a muscle cell, smooth muscle cell. This is one reason why the parasympathetic system causes that smooth muscle cell to relax. It removes calcium, moving it back into the sarcoplasmic reticulum. With less calcium, less binding sites are exposed on the actin. Okay, it's just the backwards. It's how a skeletal muscle cell relaxes. We're just inducing that with nitric oxide. The other thing that it does is it changes the phosphate head. Come on. Well, that's what I wrote there. It takes phosphate off the myosin. Remember I said protein kinases usually add phosphate? This one removes it. And without the phosphate, the myosin doesn't have the energy to attach to actin. It doesn't have the energy to change its shape. So for these two things, moving calcium and removing phosphate, myosin and actin can't interact. And when myosin can't pull on actin, the muscle cell relaxes. When the muscle cell of the blood vessel relaxes, the blood vessel dilates. When the blood vessel dilates, it compresses the veins so blood can't leave the penis, and the penis becomes swollen with blood. And that's erection. Okay? So although this looks very complicated, it's not. Okay? Nitric oxide activates guanylyl cyclase, causing cyclic GMP to form. As a result of cyclic GMP, we have protein kinases activated. We move calcium back into its storage site, and we take the phosphate off myosin, muscle cell relaxes. Okay? So that's the biochemistry, that's the physiology of erection. Okay? So now let's move to emission. What did I say emission was? Two things. Movement of sperm through the vas deferens, through the ejaculatory duct, and into the prostatic urethra, and the secretion of our three glands. Seminal vesicle, prostate, and the bulbourethral gland. That is sympathetic, okay? So this pathway, we'll draw on here, make this blank. So now we're gonna look at, come on, one, blank. So now we're gonna look at sympathetic. Trying to make it brighter in here. There we go. So let's change that word to emission. Still going to use our same sensory component. So we're still going to have stimulation of the sensory neurons in the glans penis, forming the dorsal nerve of the penis, going in with joining other neurons to form the pudendal nerve, traveling through the ventral rami to end up in the posterior gray horn of the sacral spinal cord. But there are no sympathetic neurons there, right? So it has to go up to the lumbar region. So it's going to do this by crossing over and going up, going into that sympathetic region. It's in the spinal cord, so let me draw it in the spinal cord on outside. Crossing over and going up the spinal cord. Whereas this one's going to cross over and go up this side of the spinal cord, which is where I need it. Now, some of them aren't going to stop there, right? Because you can, well, I can't, but for the glans clitoris, yes. So most guys and women can feel the stimulation of the glans clitoris and the glans penis unless your spinal cord is severed. So some of these neurons are actually going to go all the way up to the brain, all right? But for activation of the sympathetic nervous system, they have to stop at the lumbar spinal cord. This is why someone who has a spinal cord transection can still make babies, okay, as long as it's above L1. They may not have the sensory feeling, all right, processing of the stimulation of the glans penis, but their partner can still stimulate it. They can still have erection and emission and ejaculation, okay, because of this can get up high enough. All right, so now we have our sympathetic preganglionic neurons in the lateral gray horn are being activated by this sensory pathway. They're going to come out the ventral root because they're motor. They're going to come into the the sympathetic chain ganglia. They're going to come all the way down into the sacral spinal cord. And they're going to come out the sacral splanchnic nerve. Okay? Parasympathetic is in the pelvic splanchnic nerve, PS for parasympathetic. Sympathetic neurons are in the sacral splanchnic nerve. And then in the pelvis, they're going to synapse with postganglionic sympathetic neurons that go to the seminal vesicle. They're going to synapse with postganglionic sympathetic neurons that go to the prostate gland. Postganglionic sympathetic neurons that go to the bulbourethral gland. And emission also includes yours, the guy's epididymis. So, going to the epididymis, okay? Neurotransmitter of the sympathetic nervous system is norepinephrine. So norepinephrine is going to cause the vas deferens to undergo peristalsis. Norepinephrine is going to cause the glands to secrete. Okay? So that is emission. Ejaculation is all three. We're not going to look at any physiology. You don't have to worry about anything happening inside the cell. Okay? So, we're going to use sympathetic and parasympathetic and voluntary neurons. So, I'm going to use the screen picture for that, and by the time I erase all this, I think you would understand our pathway now. Ejaculation is the movement of the sperm through the membranous urethra and out the spongy urethra. Period. Okay? Primarily, that is the role of our bulbospongiosus muscle and our ischiocavernosus muscle. So it's primarily voluntary neurons, acting as a reflex though. Okay, just like tapping your knee, you kick your foot out. That's a voluntary muscle reflex. So back again to our dorsal nerve of the penis, and again, this is all written up here. Sensory information through the pudendal nerve, back to our sacral spinal cord. Lower motor neurons, voluntary motor neurons in the anterior gray horn, come out through the ventral root, into the ventral ramus, down the pudendal nerve, and they're going to innervate the they're going to relax the external urethral sphincter, innervate the bulbospongiosus and ischiocavernosus. And that gives a rhythmic contractile feeling, okay, of orgasm. Now, we also use, so that's the primary thing. So let's do that one more time. Sensory innervation through dorsal nerve, through pudendal nerve to sacral spinal cord. Synapses with the anterior gray horn neuron, our lower motor neuron. One neuron coming through the ventral root, ventral ramus, out the pudendal nerve, going to bulbospongiosis and ischiocavernosus. All right? Well, is the urinary system activated by sympathetic or parasympathetic when you go to the bathroom? Para. Normal body function. So, for the smooth muscle of the urethra, we're going to activate that with sympathetic or parasympathetic? Parasympathetic, because it's part of the urinary system. So, parasympathetic preganglionic fibers, and these are a different color, I apologize. Parasympathetic preganglionic fibers come out just like they did for erection, come through the pelvic splanchnic nerves, and then the cavernous nerves go to the urethra and cause peristaltic movement of the urethra. Also for the movement of sperm. All right. Sympathetic does the opposite of the parasympathetic for urinary system, right? We don't want any urine in the bladder to come down through the urethra while it has sperm in it. So sympathetic is going to come down and go to the internal urethral sphincter and keep it constricted. So we don't add any urine to the urethra, okay? So primarily, bulbospongiosis and ischiocavernosus muscles contract because of motor in the pudendal. Secondarily, parasympathetic causes the urethra to do peristalsis because that's smooth muscle. And then finally, sympathetic keeps the internal urethral sphincter contracted so urine doesn't go through, okay? And that's all summarized up here. Now, for the lecture exam, I do one of two things. It has anywhere from 8 to 10 extra credit points on it. Sometimes I just ask extra questions and divide by a number fewer than I asked. Sometimes I give specific structures and I say this will be your extra credit question, okay? So your extra credit question for the upcoming exam in 2 weeks is going to be one of two components, although one has 3 parts. So remember those activities that you did on the lab report for the endocrine system, where I had you identify what number 1 was and number 2 and number 3 and number 4. So it'll either be a diagram of that, probably the phospholipase C. I don't think I can get 10 questions out of the other one, so I know stuff. Or it'll be one of these diagrams. I never ask students to draw me pictures, okay? I learned my lesson. So this will be drawn for you, and I'll either ask you to draw in the neurons, or I'll have them already drawn and ask you to identify whether we're looking at ejaculation or erection or emission, and to name the neuron. Okay. So one of those 2 diagrams, I obviously haven't decided which, one of those 2 sets of diagrams will be the extra credit. Okay. So you'll be writing down the name of the molecule of its endocrine, or you'll be drawing and or writing down the name of the neuron, if it's one of these, and tell me which process it is. Okay? All right. I'm going to push through rather than take another break because we only have about 20 minutes left on the next concept. I apologize. We missed a day on Monday last week, so I'm still trying to catch up, although Thursday night did go through all of this. So, okay. Take a deep breath, stand up in your chair and stretch if you want to. I could do a 10-minute break, but I think we can push through this. You just need to don't clear your brains, clarify your brains. So these 2 diagrams, both are showing the same event, okay? So we have, I like the pictures better. I just had to correct this one, whereas this one is already correct, okay? So I wanted to point that out to you. So we have released from the interstitial cells of Leydig, or just the interstitial cells, or just the Leydig cells, they're making testosterone, which is kind of in the middle of what we're going to be talking about. But testosterone is going to go to our Sertoli cells. It's also going to go to the hypothalamus. We drew that on the second night of class when we talked about the cycle of testosterone or estrogen to the hypothalamus to the anterior pituitary. What would happen if we saw a tumor at one place or the other? So, testosterone has a negative feedback effect on the hypothalamus. High levels of testosterone causes less GnRH to be produced. Sound like an answer to the last quiz question? So GN stands for gonadotropic releasing hormone, all right? So if there's high levels of testosterone, less GnRH is made. And that's what we see here, negative feedback on the hypothalamus. Less GnRH means less LH. However, if there's low levels of testosterone, then the hypothalamus makes more GnRH. So that's what we mean by negative feedback. GnRH leaves the capillary bed 2 in the anterior pituitary and causes the anterior pituitary to make LH and to make FSH. Same thing here. LH goes to the interstitial cells. FSH goes to the Sertoli cells. Both of these terms, LH and FSH, come from female physiology, luteinizing and follicle stimulating, okay? But be that as it may. So LH binds to interstitial cells, causes it to make testosterone. FSH binds to the Sertoli cells and causes it to make androgen binding protein. The androgen it's binding is testosterone. So testosterone is going to get in the blood, but androgen binding protein from the Sertoli cells keeps high levels of testosterone in the seminiferous tubules for the developing sperm, okay? The other molecule made under the influence of FSH is inhibin. We're not going to really talk about the effect of inhibin, but it inhibits. It's a negative feedback, but at the anterior pituitary level, okay? That's where I had to correct this slide. This image actually had the negative feedback being at the hypothalamus. So I erased that and brought it to the anterior pituitary. So testosterone's negative feedback is at the level of hypothalamus. Inhibin's negative feedback is at the level of the anterior pituitary. So let's look at GnRH, okay? GnRH is released when testosterone binds to neurons in the hypothalamus, and you don't have to memorize these terms, this is the arcuate nucleus, and causes them to release a chemical called kisspeptin. Kissing is part of the hypothalamus's activity. That molecule causes the hypothalamus to make GnRH. That's all I want you to know about kisspeptin, okay? So it's an intermediate step between low levels of testosterone. Doesn't directly cause these cells to make GnRH, but causes them to make kisspeptin, and that causes the GnRH to be made. That goes through the portal veins to the pituitary, leaves the pituitary via catbed 2, and binds to cells in the anterior pituitary via a G-coupled protein, the GnRH receptor. All right? So that's what we're going to look at next. So GnRH is a short polypeptide. It's not a glycoprotein. And it gets into the capillary bed 1 through the portal veins, leaves capillary bed 2. This is a review of what we talked about the first week. I'm not going to worry about this one. You don't have to, that really happens more in the female. But I do want you to know this. GnRH binds to a G protein coupled receptor that uses the phospholipase C pathway. Okay? To be honest, most of the cells use the phospholipase C pathway. Unfortunately, some of them use the other. So I used to have a lab activity to ask which pathway, and it didn't work because you looked it up and it gave them both. We're just going to stick with this one, okay? Because it's primarily phospholipase C. So GnRH binds to the anterior pituitary, activates the alpha subunit to activate phospholipase C, which takes PIP2, phospholinositol phosphate, this is a review here, splits it into diacylglycerol and IP3, okay, and activates protein kinases and some genes, and we get testosterone production. So phospholipase C in the anterior pituitary. Now we're ready to follow the testosterone. We'll look at this next week because it's where we tie in the homologous structures between male and female. So in the male, LH is going to bind to the interstitial cells of Leydig. FSH is going to bind to the Sertoli cells. And we'll look at this slide again to see the homology between male and female. We're not going to worry about it now. So, LH binding to the interstitial cells, okay, binding to these interstitial cells, now is going to use adenylyl cyclase. So the anterior pituitary cells making testosterone do so because of the role of phospholipase C. The Leydig cells do so via cyclic AMP. We're using both pathways. One's in the pituitary because of GnRH. One's in the Leydig cells and the testes using cyclic AMP. Okay? Testosterone itself is based from what molecule?
Cholesterol.
Cholesterol. All right, anybody remember from Bio 430, what is the main organelle that is visualized in electron micrographs of cells that take cholesterol and make steroid hormones?
Smoothie gun?
Smoothie R, okay? Do you remember, and I would be shocked if you did, do you remember what the mitochondria look like in cells that have a lot of smoothie R? Typically, our mitochondria have their cristae looking like this, that inner membrane, okay? Mitochondria in cells that make cholesterol induce hormones have tubular cristae. So when we look at them in cross section, they actually look like they have smoothie R inside of them. So this is what the mitochondria look like in cells that use a lot of smoothie R. Because that mitochondria is one of the steps that we use in making that steroid hormone. So here's the LH binding to its G protein coupled receptor, taking ATP and converting it to cyclic AMP. All right? Cyclic AMP is now going to activate enzymes that regulate the rest of the pathway. One is to take cholesterol. The cholesterol is sent into the mitochondria, and the cholesterol is converted to a molecule known as pregnenolone. So cholesterol actually I should draw it round because in these cells it is round, unlike what they show there. So cholesterol is converted to and now this goes into the smoothie R. Okay? And in the smoothie R, it's converted to a couple of different things. It can go to progesterone and then go to testosterone. But essentially, it's going to go to a molecule called androstenedione. And then this is going to be eventually converted to testosterone. Okay? And that's happening in the smoothie R. There is an enzyme present in the Leydig cells that will take testosterone and convert it to DHT. Anybody heard of DHT? Baldness, male pattern baldness. So this stands for, where is it here? Dehydrotestosterone. And it's a secondary sex characteristic hormone, okay? So this is going to result in beards, dense bone, a lot of muscle tissue, all those things we think of when somebody looks like a guy, okay? So remember I talked about that island where the guys look like girls until they reach puberty? This is what they're lacking, okay? They don't have enough of the dehydrotestosterone to cause that to happen in the fetus. So secondary sex characteristics, we're talking the phenotype of a male. And the enzyme that they don't have enough of, we're almost done here if you're getting sleepy. The enzyme that they don't have enough of is called alpha or 5-alpha, either way is fine, reductase. Okay, that's the enzyme that converts testosterone to dehydrotestosterone. So we need both the mitochondria and the smooth ER to do this. By the way, when we get to female next week, we're going to get to female on Wednesday, but when we get to this process in the female, same thing occurs. Women make testosterone. All right? They just don't make very much of it. But they make enough that when they reach menopause and they're not making very much estrogen, they're making even less to no testosterone, and it can affect some of their sexual libido. Okay, because it's testosterone that's required for that. All right, so now we're just going to spend one slide, maybe two, on the Sertoli cells. So this is LH going to the Leydig cells. There's a lot of stuff going on here. This is what I want you to know. All right, so just because this is on a PowerPoint slide doesn't mean that you have to know every single step in there. All right. This is a diagram you have already in the PowerPoints. I just kind of drew it myself. So LH binds to its G-coupled protein. We're still talking about the Leydig cells. The adenylyl cyclase becomes activated, converts ATP to cyclic AMP. Cholesterol is converted to pregnenolone in the mitochondria, and in the smooth ER, that's converted to testosterone. Okay. All right. So everything there. So this is what testosterone does around the Sertoli cells. It binds to the protein made by the Sertoli cells, and it's going to primarily be responsible. This is the one function here I want you to know, although the others are secondary. It's spermiogenesis. Remember how the sperm changed shape from early spermatid to spermatozoa? That's under control of the testosterone. All right, FSH binds to Sertoli cells. It's also going to use cyclic AMP. Okay, so cyclic AMP is going to be both the Sertoli cells and the interstitial cells. Here, the main thing is androgen binding protein, inhibin, and I'm going to add to that estrogen. That's not an important thing, but I want you to know about the estrogen, okay? So, let me just write the one, erase this. I'm just going to write one chemical reaction, all right? But I want to make sure you have this down, because I want you to know one more enzyme. So the Sertoli cell does not take cholesterol and convert it to anything, but it makes estrogen. All right? So the Sertoli cell, although or sustentacular cell, with the nucleus up there so you can remember it, is going to have a G protein coupled receptor, or FSH. And FSH is going to cause it to make androgen binding protein. And inhibin. These are not cholesterol based. All right, so they're going to go through the ribosomal ER and all of that. But the testosterone coming from the Sertoli cells, remember, it's lipid soluble because it's cholesterol based. So testosterone passes through the plasma membrane because it's cholesterol based and it's soluble in the plasma membrane. And in the plasma membrane, a small amount of it, because most of it stays out here in the lumen, binding to the androgen binding protein. Testosterone is going to be converted to estrogen, a small amount. So just like women make a small amount of testosterone, men make a small amount of estrogen. And the enzyme here is aromatase. Okay? So, as a preview for next Monday's lecture, let's go back to this picture right here. So in the male, LH binds to Leydig cells, and they make testosterone. Testosterone goes into the Sertoli cells, and they make estrogen. That's what this little E is here. Okay, that's what we just kind of summarized. In the female, LH goes to the cells around the outside of the follicle, and they make testosterone, or the early form of testosterone. That goes into the follicular cells around the egg, and they convert that to lots and lots of estrogen. So both cells are doing the same thing, with the same enzyme. It's just different amounts. The male makes a lot of testosterone, a little bit of estrogen. The female makes a lot of testosterone, but converts most of it to estrogen. So only a small amount of testosterone is left over. So we'll review that on Monday. Okay, we're not going to talk about it Wednesday. We'll review that on Monday. Okay, we're done with lecture. Okay? I think that's so loud. So, being done with the male reproductive, you have a lab quiz on Wednesday. So, that won't include any of this physiology stuff. All right? It will be models and histology. So, naming the endocrine organ models, identifying the endocrine histology that I asked you to look at, which I believe was anterior, posterior pituitary, thyroid, parathyroid, pancreas, and adrenal gland. So just naming the organ, okay? With pituitary, you need to be able to differentiate between anterior and posterior. You don't have to name any other parts of the other tissues except the organ, okay? All right, with the male, now most of you looked at the testes last week. So tonight we have the epididymis, vas deferens, seminal vesicle, prostate gland, and penis. Okay? You don't have to identify any structure within the microscope slide, just name the reproductive structure or organ. Okay? We're done with by done, I mean we finished all the components of the male reproductive pathway. So most of you looked at structures of the scrotal sac, the cavernous muscles, the vas deferens. You may have already looked at seminal vesicle, all right? But now with the penis, you can add the corpus cavernosa and the corpus spongiosum, the muscles. Just as a heads up, I don't want to knock down these models here. I mean the bones. It's okay. So if I tilt this, it'll be hard to pull this off, but you're welcome to do so. Sorry, penis. So running along this area right here, that yellow structure that you see is the pudendal nerve. Okay, on its way back to the pelvic cavity. So there's it's, the dorsal nerve is going to be on that poor penis that I broke off, but you can't see it. So the pudendal nerve that you can see in this cavity. Alrighty. So that's the rest of lab time. I'll pull out the male reproductive components over there. If you don't find, if you want to look at endocrine, they're over here. But for now, I'm going to focus on the male components. And I'll pull out the other models that deal with that. All right?